Welcome to the Phase Space Invaders podcast. I'm happy you're with us for another episode. It's time for conversation number 18, and my today's guest is Erik Lindahl, Professor of Biophysics at the University of Stockholm and KTH Royal Institute of Technology and SciLife Lab. Many of you will know Erik as the coordinator of the GROMACS project, but this is far from all he's been doing these days. In fact, his group not only develops and applies computational methods to study pentameric ligand-gated ion channels, but also has an experimental branch dedicated to structural biology, which means they can approach biological questions in the field of neuronal signal transmission and neurotransmitter function across many techniques and scales. And so accordingly, Erik defies the standard mindset of a computational scientist by reminding us that despite our dependence on computational power and advanced technology, real breakthroughs are often waiting for these who have the patience to think carefully, come up with eye-opening ideas, and follow their sense of purpose. We discuss the different ways to be smart in science, highlighting the paradoxical need for both complexity and simplicity in thinking, and talk about what kind of questions in biology will keep us all busy for decades to come. Finally, Erik shares the story behind his series of lectures on concepts in molecular biophysics, one that I absolutely recommend if you haven't watched it yet. And they are all available on Erik's YouTube channel. Hope you enjoy our conversation. So, Erik Lindahl, welcome to the podcast.
Good morning, Milos. Great to be here.
So, Erik, I believe by now your name universally evokes an association with the GROMACS software, but you're also deeply involved in regular research, primarily in the field of membrane receptors and channels. And with these two sources of practical insight, I wonder how you see the future of the cycle of discovery in molecular biophysics. Will it be mostly driven by new experimental data or by new software or hardware, or maybe we're just still mostly bound by human imagination?
Well, that's a good question. At first, in the defense, not just of my team, but there are many groups all over the world doing tremendous software work. So I think that software work today is a normal part of main biophysics research. But overall, I feel that I think the older I got, and in particular some 15 years ago when we started doing our own experimental work, I've realized that we frequently care a lot about techniques, methods, and everything, which certainly is important. But the really hard part here is focusing on solving fundamental biological problems and using both computers, but also a number of experimental techniques to make it faster. And I think biophysics as a field, not just the computational part, has made tremendous advances there. We've seen free electron lasers, cryo-electron microscopy, amazing super resolution microscopy. So this whole traditional division between, on the one hand, experimental work, on the other hand, computational work that existed some 30 years ago when I started the field has completely disappeared. Biophysics today is a data-focused research ranging from original collection of data to very advanced processing and data-focused work where we learn from it, which is fantastic. It's a great field to be right in the middle of and trying to keep up with much younger people.
I see. So would you say we're ready to incorporate all those streams of data, all those experimental techniques into our simulational techniques?
I'm not sure whether that should be the goal, right? That, as I mentioned originally, that there are many things that are amazing with all the computational power we have today, that you can throw more data at an algorithm than we could even imagine some 20 years ago. But I feel that we frequently forget this part, that the most important part about science is thinking about a problem that is actually not yet solved. We do not know the answer, and then finding ways to solve that problem. Now, in some cases, data-driven methods might be a good way to do that, or simulations. But in a surprising number of methods, that might be an experimental approach, or simply sitting down with paper and pen and thinking. I think we spend too little time thinking and too much time occasionally using computers as a brain prosthesis.
So you would say we're on the imagination side where the effort is going to lie, right?
I think that It's been that way for 40, 50 years in biophysics, and I think it's going to remain that way for 50 years. Having smart ideas is always the biggest bottleneck, which I think is great and refreshing in so many ways, right? Because those great ideas does not require you to have access to, say, 1,000 GPUs or a whole lot of these fancy resources that we might have at some fairly rich institutions. No matter where you are in the world, the human capital is fairly evenly divided, and you can have a great idea even if you don't have access to tremendous and very expensive resources.
Okay, that's an interesting perspective. I haven't thought about this kind of timeless versus time-dependent research agenda, right? Like how much of your ideas can be thought of at any time in history versus how much you're actually contingent on the current moment in the history of research. So the word vision, is this something you would propose as something that propels research?
I'm not sure. I think it's a good... Helmut Schmidt, old German chancellor, there's a saying, it's probably not true, but he used to say that anybody who has visions should see a psychiatrist. Visions can be good. I think it's good to have an idea where you're heading. Grand visions, I think it's a good idea to have a personal vision. Why are you doing what you're doing? What is the reason why you're doing it? Are you trying to understand protein structure or understanding the nervous system? There are many problems that are great. Have a reason why you're doing it. The mere fact that something is faster or slightly better is usually not a good idea. But why should it be faster? Why should it be better? How will this help us, the world, and the future generations to understand biophysics and biology better? In some way, that might be a vision. But then on the other hand, there are some great method development team, and they see their vision is to enable others to solve these problems. And I think that's equally valid. If I say, somebody's training a machine learning method, and I know I'm doing this precisely because I want to allow people to, say, classify cat pictures on the internet better. Now, if that's your goal, more power to you. But just be sure why you're doing something and make sure that you're doing something you're enjoying.
Okay, so that's a multitude of voices approach. Everyone finding their own vocation or calling and following it. What is yours? What is your vision then? What keeps you up at night, in terms of problems in biology that you want to solve in the next years?
Yeah, in one way, I'm not sure whether I'm an exception to it, but I've always had two mistresses in life. And these mistresses, at least in the professional life, one of them is computers and the other one is biology. And the hardest decision of my life was deciding whether I should go into physics or medical school. And I thought I made that decision in the mid-90s when I went into physics. The only problem is that biology didn't agree, and it has kept pulling me back for the last quarter of a century. So more or less I mistake. I've ended up in this situation where on the one hand, I'm working on life science problems, but we're very much doing it with physics and particularly computer-based solutions. Over the last 20 years or so, I've fallen in love with ligand-gated ion channels, in particular the ion channels in the nervous system and understanding neuronal transmission. For two reasons, because on the one hand, it's a very physical problem. You're literally conducting a signal in our body, right? There are a few things that are more physical than that. But on the other hand, it's also an inherently chemical problem. Now, if you're listening to this podcast on a Friday, you might go out and have a glass of wine, and those ethanol molecules are directly modulating the conductance across a so-called synaptic cleft. So that we have a multitude of ways where both the environment or drugs and anything can fine-tune the signaling. And I simply find it amazing how nature has been able to evolve and optimize this over billions of years to create both very highly selective signaling, sometimes protect ourselves from the signaling. You might have heard there are these, for instance, these toads that are highly poisonous that people use. The Indians in South America would take the arrows and use these toads to add poison to their arrows. The whole point, why doesn't the toad die from this? Because this is a poison that then would block these channels. It turns out that there are a handful of mutations in the binding site that make sure that this particular ligand-gated ion channel in the toad is not sensitive to the poison produced by the toad. And this, of course, we all know why this works. It's natural selection, right? But the fact that we managed to combine physics, structure, selectivity, I know how it works, but also on an emotional level, I'm still flabbergasted that it actually does. And that keeps paying back. And I hesitated a lot when we started to do experiments in the team, but in hindsight, it's probably the best decision ever, because some About 15 years ago, I felt that led to an entire second phase of this lab, that we can producing our own data and we can start to study the problems directly rather than just relying on data produced by others. So that I still identify myself as a computational group, but we're also very much an experimental group nowadays, so that we love all these techniques no matter what they do. The only thing that's relevant to us is this is a technique that can help us understand these ligand-gated ion channels. That could be SANS, it could be cryo-EM, X-ray crystallography, you name it. And if it's a technique that doesn't do that, it doesn't really help if it's the world's best technique, if it's not helping me solve my problem.
Right. As a curiosity question, how hard it is for someone with an established computational career to start an experimental lab as a separate entity?
Well, I had done experience as an undergraduate, not a PhD student. I think anything in life is difficult, but you need to recruit people who can help you do this. And I have a tremendous team here in Stockholm. Rebecca Howard leads the experimental team. We have fantastic postdocs and the PhD students. No one mentioned and none forgotten. But I don't think that's fundamentally different from computational work. Nobody in the world will be an expert in every single computational method, and you likely can recruit people who have expertise in various computational methods in the team. And good teams are usually teams where you allow people to have different expertise. And I think our role as PIs, team leads, no matter what you call us, is to really explore this, make sure that we recruit people with diverse experiences and get them working efficiently together as a team.
Right, because I think a lot of people will hesitate to do that, right? With the load of work that we usually have, to accept a bunch of new knowledge to internalize and new people to work with in a field that you don't really know that well. It is a big bet that I guess it pays off in the long term, right? But in the moment, it might be a really daunting challenge.
I know that I've thought that too. In hindsight, I'm not sure where we're thinking the right way, because nowadays my team is big, so do as I say, not as I do, etc. But I think many of us have, as we develop large teams, we become ocean liners that get very difficult to turn around and set in a new direction, if nothing else, because you might have 5 students in a pipeline. If we think of ourselves more as individual scientists, previously in our careers, we've usually been pretty good at approaching different problems, learning things at university. I think there are a few students on the master level who find it difficult to learn new things. That's why we are in university. And that's just as a new computational method is difficult, cryo-EM is difficult, but of course, every single method, new methods, when they appear, they are new by definition. Nobody is an expert on this technique yet, possibly apart from the person who has developed the technique. And I think we need to get rid of this idea that we should not do things that are new or difficult. Those are exactly the things we should do instead. Now, the question is, how do we find the motivation there? In particular, those first few years when it's difficult and we don't consider ourselves experts. That's where I think, for me at least, it helps to focus on this. Why am I using it? We didn't start doing cryo-EM because we were the best team in the world of doing cryo-EM, pretty much the opposite. But of course, we had a need for cryo-EM. It was potentially a revolutionary technique that could help us determine different states of these channels we were interested in, even if we didn't have crystals from them. Now, that's exceptionally valuable. And that value is likely worth spending a few years, even though we weren't structural biology experts at the time. Well, maybe Reba, but not the rest of us. And of course, after a few years, in some techniques, you're less lucky. Some techniques, you're more lucky. And at some point, we all choose to go after the directions where we have more luck or where the results look better. And at least in my team, I think that's every single new technique we picked up. That's how we've done it. There are some techniques we've abandoned, not because they're bad or not because we were bad at it, but they didn't pay off quite as much as something else. So that there is this balance of dare to explore the unknown, but at some point realize when it's time to not necessarily give up, but also go with your winners. But we should do more new things.
Absolutely. I think this is a consistent message that's sounded on this podcast. Yeah, so that also bleeds into another passion of yours, famously, which is data, right? You mentioned that, we have a lot of sources of data, but how much we can trust these sources of data is a big, big question. So I understand that having your own source, controlling the uncertainties and the pipelines is a big advantage. But do we also have ways of addressing this on a systematic level, like working with databases, sharing data, metadata, and so on?
I think that's critical. So first, I wouldn't say that we can control things by producing our own data, but it's probably rather that we produce our own uncertainties. I'm not sure whether that is better than somebody else's uncertainties, but doing experiments yourself certainly gives you another type of respect for experimentalists. I'm not sure it's necessarily given me more respect for experiments, because experiments is just as noisy and complicated, occasionally ugly as computational methods can be. They're just ugly in different ways. The challenge with data is always quality, right? What data can we trust? And I think as a community, we are improving. I think in general, the computational community is mostly ahead of experimental ones. The exception in the experimental communities are likely historically the data sets that have been very expensive or difficult to produce, starting with X-ray, genome sequencing. We're seeing with cryo-EM now that we're really good at assembling both raw images and the actual electron densities, as well as the structures in the PDB. If you look at small angle neutron scattering, they have these fantastic databases where if we go down to one of these facilities to record a spectrum, it's not just that the spectrum are put in databases. So if my student and lab manager go down there for recording, it's really cool. You get a DOI for the experiment. So this DOI includes the student and the lab manager from my team who went down. It includes the beamline scientists. It does not include me. And that might sound strange, but at that point, I was not physically there to conduct the experiment. So this DOI is specifically for the experimental data. And I think it makes a ton of sense. And it gets even cooler when you realize that you can have up to 6 months embargo. But then automatically, everything is available after 6 months, no matter whether we have published or not.
DOI, by the way, can you explain the abbreviation?
For a digital object identifier. So it's the idea so that you can find it in a database. And
right.
Effectively, this, of course, counts. It is a publication. You can track the data. You can assemble this data in many ways. In life science, I don't think we're quite there yet that we can force all cryo-EM experiments and everything to be public after 6 months, whether you published or not. But we're also taxpayers, right? And as a taxpayer, if we are paying for these facilities, we're paying for somebody to collect their data. I'm paying for this data to be collected. I'm not necessarily paying for somebody to limit access to the data to benefit their career. And this is, of course, a cultural tectonic shift. I think it is happening, but it's likely going to be up to the next generation to drive this even more. Now, if we translate this to, say, molecular dynamics or anything, should we collect molecular dynamics data? Had you asked me 10 years ago, I would have sighed and say no, because there is so much crap data out there. And I think that's still true. There is a lot of crap data out there. Now, to be honest, there is a lot of crap experimental data out there too. The cool thing in experiments, we have learned to handle this. Not by disallowing people to deposit some data, but being better at quality indicators. So if you have a PDB structure, anybody can upload a structure to the PDB. But of course, there will have to be quality assessment reports and everything. So if I go and check these structures, I will see that particular structure from Erik Lindahl is crap, and then I will likely avoid relying on it. I think the key to MD, AI, whatever data we imagine, is that we need to get better at developing quality assessment indicators so that out of these structures, I know what can I trust, how much can I trust it. And I hope that could have tremendously important ways to actually, not just short-term to me as a consumer, decide what structures I use, but long-term, we're all also producers, right? And if I get these quality assessment reports when I upload data, it will hopefully start to drive quality improvements in my team, that we get better at producing data that fulfills high levels of quality. So what can we do if all this data is available? Well, today, the obvious consumer here is, of course, various machine learning methods, right? In general, I would probably prefer to train things on experimental data when I can. The challenge is that producing new experimental structures, and in particular any type with motion resolved or so, is filthy expensive. While molecular dynamics simulations, if we, for a second, trust the experiment slightly more, even if the molecular dynamics simulations or AI models are not quite as accurate, they are probably a factor 1,000 cheaper. So at least where AI is heading today, having access to factor 1,000 more data is probably in some cases more important than having the highest possible quality data.
Right. These are great points. I'm just sometimes thinking, what is the extra information that lies in the dynamics, right? Because a lot of simulations, equilibrium simulations in particular, will not really explore much more than just fluctuations around the ground state, around the native structure, right? So how do we even combine things like enhanced sampling and steered MD and simulations that sample multiple conformations, especially when they don't keep track of the underlying physics in a strict way, like with free energies?
I'm a bit allergic to the word dynamics per se, right? Because I think you're quite right. The dynamics per se does not add much. If anybody has read chapter 1 of the GROMACS manual, I think we all said that, we're not doing molecular dynamics and Newton's equations of motion because it's some sort of super fancy dynamics per se. It's just a really efficient way for all atom systems in particular to sample the entire phase space. And of course, if you know the entire energy landscape of a molecule, we know everything that we need to know about the molecule, and then we can derive the dynamics from there. I don't think there is any, at least not a lot of inherent value of simulating the exact dynamic during the simulation, because remember that we do not know the exact initial conditions anyway. So that dynamics is just one or a handful of examples of potential trajectories. This is potentially also what makes AI methods so interesting, that it's not the dynamics per se we're getting from molecular dynamics. They're efficient ways of exploring the phase space. And although if AI methods might not quite be there yet, at least not for large molecules, I'm quite confident that they will be able to sample phase space quite efficiently long term. And if they can do that more efficiently than what we're doing with current methods, they're going to be just as efficient to determining the entire energy landscape. And then they're just as good for understanding the dynamics.
Right. That brings us back to the question of receptors and channels. Is your perspective that, we understand them in principle and it's now just a question of engineering and data to collect all the interesting biological facts about them? Or is there something that you would inherently say we don't still understand or grasp about how these molecules work?
Well, first, there are numerous families of receptors, right? And there are a bunch of them that are, well, not completely unknown, but mostly unknown. So they're going to be work remaining here for generations of scientists to come. I think simulation groups in particular, or maybe my generation in particular, we tended to pick things where the structure was already known. With AlphaFold and other things available today, that we don't necessarily have to start with things where we already have a PDB structures. It's much easier today to pick a white spot on the map and start using computational methods to explore something where we don't even know the structure yet. But if we limit ourselves to my receptors, the ligand-gated ion channels, you could argue that on a very fundamental level, we understand what happens. It's a membrane protein where the channel goes to the membrane. There is a domain outside the membrane, an extracellular domain that binds the ligand, the neurotransmitters. And when these bind, the receptor undergoes some sort of protein earthquake, and the channel pore some 50 angstroms away magically opens. And that we know. But the devil in the detail here is that how does it open? Biologically, all these receptors, they only open transiently, which makes a lot of biological sense, because that producing the ion imbalance, which we do with the ATP pump, is one of the most expensive processes in human cells. Well, that's why bacteria, for instance, don't have a nervous system. But once you're opening the challenge to create these signals, the problem is the ligand is bound, right? And the ligand is going to be stay bound. That's the whole definition of bound. It likes to be there. You don't want these channels to stay open for a second and keep leaking ions. It would be very expensive, but it will also mean that we couldn't really create new signal in the nervous system. So these sensors, they undergo a process we call desensitization, that in a millisecond or so, they collapse and stop conducting. Where just the last 2-3 years, we're starting to get the point where we're understanding these processes. And we still don't understand the process. How do we go from desensitization back to the resting state? How do we recycle so that we can go through an entire cycle of these receptors from resting to transiently activated to desensitized and then charge back? And to us, what's tempting with this is all the various drugs by which we can modulate this process, and even more so that This is difficult in our lab when we're working with simple model channels. Our simple model channels are so-called heteropentameres. I didn't tell you that these channels consist of 5 subunits. In many cases, we and other groups, in particular on the computational side, work with 5 identical subunits. But biologically, most of the biologically interesting receptors are heteropentameres. They consist of at least 2, if most commonly 3, and sometimes 4 different subunits out of 19 genes. And this creates a remarkable diversity of cooperativity or independent motions and everything that, of course, have very important natural selection effects. This is likely what gives rise to the entire diversity and signaling in our brains and everything. And we understand almost nothing about it. It's going to be decades of work remaining just for this single family of receptor. And that's so great with biology. There are hundreds of them remaining after that one. It's not going to end with my life.
I see. That's amazing. I think a lot of the answers on the complexity side will have the form of, oh, here's the data that answers your 100 particular questions, right? But how do you even synthesize an insight like this? Like, as what you say about the heteropentameric channels, is there an abstraction that you can make of the data and hold in your mind as an answer to, oh, what is the role of heteropentameric ligand-gated channels in general, right?
I think computations serve one important tool. There are things we can do with computation that are very difficult to do in experiments, in particular accessing these transient states, what's actually happening during opening and collapse processes, and how are the different subunits behaving. And remember that these are transient states that are very difficult to capture experimentally, and if we can't capture them experimentally, it's very difficult to understand the structure. Now, having said that, simulations, certainly, they have the limited accuracy. Many of these channels are remarkably sensitive to the lipid composition in the membranes. The human ones don't really work efficiently without cholesterol, for instance. So to us, one of the most powerful approaches has been to combine simulations with so-called electrophysiology experiments, which sounds complicated, but it's not really. So that means that we take DNA from a particular receptor, and then we inject a few nanoliters of this in a frog egg, Xenopus laevis, the largest egg cells that exist. They're a millimeter across. And then we put them in a fridge for 3 days, or incubator, as you say in the lab. And then a few days later, now 99% of all the membrane proteins on the surface of this small cell is going to be our particular ligand-gated ion channel. Now I can put 2 glass pipettes on this and literally measure how much current is going through this membrane when I'm subjecting the channel to different conditions, both voltages and external molecules. And these type of experiments, to me, they're the extreme opposite of simulations, right? I have no atomic detail whatsoever. I can't see anything about what happens structurally. The one thing I can see, though, is that we get absolute answers what's happening biologically. Is it increasing conductance or decreasing conductance? If when I try this with different subunit compositions, is the process happening slower or faster, more or less? And if you then, that gives you an ability to use simulations at Gedanken experiment, that we can have models, ideas, but then of course it's imperative that we actually assess this and compare it to the experimental results. If I believe that this simulation model should result in something, is this actually what I see in an experiment too? And I have a great example of that. We fooled ourselves many years ago, and we never ever published this, and that's why it can be cool to mention on a podcast that when we got started with experiments, I had this passion that we would like to show that we could do something in a simulation and then show that we could predict the experiment, and that fairly simple changes to a channel, that should be easy to do in a simulation, right? So these channels are fairly hydrophobic on the inside. So we figured that if we make this pore now more hydrophilic by replacing some threonines, I think it was leucines with serines or threonines, then I make the entire pore more hydrophilic. We did that in a simulation. The pore is definitely more hydrophilic. The pore is larger. You see a much larger radius. And then you go into the lab and expect to confirm this, that this should be open easier and have higher conductance. And what you see in the lab is the extreme opposite. It has almost no conductance at all. It is significantly harder to open the channel. And then you start wondering, what went wrong? And I realized with all these serines or threonines, what we have created was effectively an ion binding site. So that what happens is that the first ion that goes through the pore, instead of passing through the pore, it gets stuck in the pore and binds there. And this is actually known experimentally for divalent ions. And we had just created a similar site for a monovalent ion, which of course, it gets these memento moris that in a simulation, you think that you can measure conductance by the radius of the channel. But what you're actually measuring in the experiment is the number of ions per unit of time that go through. And if you try to measure the number of ions per unit of time that go through, you can do that with computational electrophysiology, right? The result matches perfectly. So I think it is important to compare simulations and experiments, but there's this analogy with René Magritte, right? Cécine Pine Pips. The simulation is a model. It's not the reality. And don't confuse your model with the reality.
Yeah, absolutely. Even with that, I think it eventually makes you humble at some point, knowing that you come up with a simple explanation and then the simulation or the experiment tells you that, oh, there's actually a very obvious alternative that you didn't consider, right? That there's a evolutionary balance of forces and you didn't think of that in the first moment, being fixated on a different flavor of the model.
Yeah, and I
think each one of us experiences that.
And I think I can say this as a computational person, if I wear my computational hat now, that some of the smartest people I know are computational researchers. And I'm so in shock and awe about Klaus Schulten when he was still around, Peter Coleman, Benoit Roux, others. And yet, the danger with us being computational, we're smart. We're smart at the physics. We know the math in and out. We know all the details about the methods. Many of these computational persons, we could likely derive the Bloch equations with NMR or so. While if I look at some of the smartest experimental colleagues I have, or rather the experimental colleagues I mostly admire, like Gunnar von Heine here in Stockholm, they frequently go for simplicity because they're so deeply aware about how insanely complicated the systems they're studying is. So the only way to understand this is to ignore all the details, go down to very simple questions that you can ask in a lab and get yes or no answers. And I think 25 years ago, the point is that is, of course, not simple at all. The hardest thing in life is learning to identify things, to drill complicated phenomena down so that they can be answered with very simple questions. And that's probably also an important lesson. Computational people like us, we are smart is probably not the right word. We're good at math, we're good at physics. Don't confuse that with intelligence. Intelligence is frequently the ability to being able to ignore all the details and just come up with one very, very smart experiment that will answer the same question without the need for all those equations.
Bottom line being, simplicity is hard. Yes.
Simplicity is hard, but it's worth to strive for.
Great. And you strive for it with, I always like to promote good resources for the community. So I just wanted to bring up your lectures that are available. Where are they available? Can you point people to? Oh, that's on
YouTube. I don't even know the link, but if you search for my name on YouTube, you would likely find them. I'm very happy that they're useful. That this was a side effect of the COVID pandemic. So that with 4 weeks notice, I for a long time, I used to record my lectures just with a built-in camera or a small camera on a tripod. But it's such a horrible experience because it's a B, C, or D class experiment compared to actually being in the room, right? And you're seeing, in best case, you're seeing the teacher's nostrils. And in worst case, you're seeing the back of the teacher when I'm writing on the board. But during COVID, we were all told with 3-4 weeks that now you would have to have complete remote teaching. And I had seen Adrienne Reutberg at the time giving a talk a few months earlier. I think I was using one of these light boards at University of Florida. And what can I say? I'm a tech geek. So I figured that if I've for a long time, I've thought about doing a proper studio version of this recording. And I tried to get my local university studio interested or something. They didn't have time. So at some point, I just bought a bunch of equipment over Christmas. And I probably spent way too many nights at 2 or 3 a.m. in the morning here in the lab. The great thing with COVID is that all the rooms here were empty. So I could steal a lecture room here in Solna and I set up a studio and I had it working for 4 weeks and I recorded 298 of these small talklets. They're roughly, I split all my lectures into 3 to 5 minute concepts where I go through all of them. And this was for my students at KTH, but I figured that if I anyway did the work 20 years ago, I would have likely focused on all the imperfections, and there are a bunch of mistakes in these lectures. But the point is, the important thing is not the mistakes, right? The important are all the things that are not the mistakes. And hopefully there are fewer mistakes than things that are right in them, at least. And at some point I decided to flip the switch and make this available to anybody. And that's the cool thing. I think there are more students that have watched these online the last 3 years than I've taught in person the rest of my career at KTH, which is a bit humbling. But I'm happy that it's useful.
Yeah, I think it's an amazing resource, and it's great to have a repository of, questions, problems in the field that go back potentially decades. People have the reference to know what is important, what is not. And yeah, I'm always happy to promote, again, a kind of unifying pictures of the fields that bring knowledge to the younger generation.
But I can just say that follow up. Don't think of yourselves as younger generation if you are, say, a PhD student listening to this. The fact that, in particular, the computational side of our field depends so much on computers, that is difficult for those of us who are elder. It leads to a remarkably fast pace, so that most of the things on the computational side, at least, that is more than 10 years old, isn't really that relevant. So that all of us have to keep learning. And I guess this podcast is another example of sharing things, right? That even if you are fairly junior, sure, you might not have my or somebody else's perspective, but you're likely more on top of, say, novel techniques or the latest thing that you're working with. So one way to contribute to this is find ways to sharing whatever you're working on right now as a student. You don't have to wait 10 or 20 years to do that. And I frequently look up resources online, and I couldn't really care less about how senior the person I'm learning from is. If they know it better than I do, I'm more than happy to learn.
Absolutely. I think there's also value in, knowing the fundamentals of free energy methods and absolute basic statistical mechanics, which is what maybe some people these days will skip in favor of, statistics heavy machine learning, right? And so there will be different accents in different generations. And I absolutely think getting inspired by different perspectives both ways is of great value. So
that's
very
true.
I try to promote. Okay. Erik Lindahl, thank you so much for the conversation, for the insights.
And thanks.
That was a great conversation.
Great joining you.
Hope you have a great day.
Good. Ciao.
Thank you for listening. See you in the next episode of Space Invaders.